We've talked some about how to search a library for your gene or protein of interest, but one thing to think about is how limiting it really is using a lot of these cloning vectors, especially those that are plasmids or even viruses. What is that major limitation? Well, it's insert size. For plasmids, you're really limited to an insert of about 10 kb, which again might be great for a cDNA library because the majority of a full-length gene, they're going to be under 10,000 base pairs. Majority, but not all. Lambda viruses are better. You're at about 20,000 base pairs. There's these type of vector called cosmids that get a little bit bigger. Then we have Bacs and Yaks that can hold anywhere from 300,000 bases all the way up to 2 million base pairs. These, as you might imagine, would be far more useful when we're talking about genomic libraries. What is a BAC? It's a bacterial artificial chromosome. It's a type of modified plasmid. It has an origin of replication, it's circular, but the way it's been created allows it to hold very large inserts from 50 to 300,000 base pairs. There are challenges. The transformation efficiency is quite low. What that means is that it's difficult to, let's say, encourage E. coli to take them up. So then what can we do? Why would we use it if it's really that challenging? Those smaller vectors are a lot easier. And I kind of alluded to this already, why we would still do this, why it would still be popular. And that's mostly because you can use it for genomic libraries or for very, very large pieces of DNA. A better name, instead of a bacterial artificial chromosome, is really a low copy number, large insert plasmid. But BAC is so much easier to say than low copy number, large insert plasmid. That's what they're trying to show you in this image. These are regular plasmids, you see, right? Some are small, some are a little bit larger. Here's a real big one. And this is supposed to represent the bacterial artificial chromosome. And then here is the bacterial chromosome itself. The average bacterial chromosome is going to be something like E. coli, 5 to 6 million base pairs. So a plasmid that can have an insert size of about 300,000 is still a lot smaller than the overall size of the chromosome. On that first slide, you also probably saw the yeast artificial chromosome, or the YAC. These can be used in eukaryotic cells, like yeast. They have a centromere, they have telomeres, they have various origins of replication throughout, just like we would see on any linear chromosome. But of course, they can have much larger pieces, 600 to 2 million base pairs. Again, this would be really, really useful when we're talking about genomic libraries, because you would need far fewer clones to ensure that you covered the entire genome. Often when we characterize these, we can use what are called southern blots. So a southern blot is just running a DNA gel. You transfer the DNA from the gel to a membrane. You use a probe you've designed to find out which clone has your gene of interest. Most commonly, the probe is from one species and the target DNA is from another. So you could look for, like on this slide, southern blots of various organisms' DNA using a radioactive probe from a gene called Antennapedia in Drosophila melanogaster. So here's Antennapedia in Drosophila melanogaster, and then we can check against beetles, chickens, and mice. And what we see is that, we can get the binding at approximately the right size, above 1. So here's Antennapedia. Here it is in beetles. Here it is in chickens. Here it is in mice. And remember, because we're starting to get more and more distantly related, when we do this type of southern blot, we might want to change our stringency. So we could lower the temperature the more distantly related we get. We could also change the salt concentration. One of the nice things about doing a southern blot, especially if you're talking about one of those really big yeast artificial chromosomes, is that you could look for multiple genes at the same time. You've got 2 million base pairs in there. Perhaps there's multiple genes that we could find. And then I like Antennapedia. It actually tells you what the gene does. It puts feet instead of antenna. So Antennapedia, feet for antennas. So Drosophila are weird. There's also a modification to a southern blot that is called a northern blot. So southern is called a southern blot because the scientist who made up the technique, his last name was Southern. And then because it was a direction, we just have direction blots now. So a northern blot is almost identical to a southern blot, except we are going to run RNA on the gel instead of DNA. So think about that. We're running RNA instead of DNA. And what does that tell us? So let's say we have cDNA libraries from a species, whatever, say it's a mouse, from all of these different tissues. We have a cDNA library from mouse muscle, from mouse liver, mouse small intestine, mouse brain, mouse fat tissue. We extract the RNA from a mouse, from those tissues, right? We run it out on the gel, and we probe. So I've extracted muscle RNA, liver RNA, small intestine RNA, brain RNA, fat RNA. I ran it out on the gel. And I have 2 genes I'm interested in, gene 1 and gene 2. I don't know where those genes are expressed, but I want to test these 5 tissues. And so if I do a northern blot, I run out my muscle RNA, liver RNA, etc. on the gel. I do my transfer. I have my probe I've designed, that 1 probe for gene 1, 1 probe for gene 2. What I've now learned about gene 1 is that it is not expressed in muscle, it is expressed in liver, it is not expressed in small intestine, it is expressed in the brain, and it is not expressed in the fat. So by testing these 5 different tissue RNAs, I've learned that gene 1 is only expressed in 2 of them. I can now go to gene 2, and I think hopefully we all can clearly see that gene 2 is expressed in all 5 tissues. The other type of blot that is very common is called a Western. And again, it's not a person named Western, it's just a direction. There is no Eastern, no set Eastern. If you look up Eastern blot, people will call it different things. So we're only going to talk about Southern, which is looking for DNA, Northern, which is looking for RNA, and Western, which is looking for protein. So in the case of a Western blot, we extract the proteins, maybe from our tissue of interest, our cell type of interest, and we separate those proteins on a gel. It's similar to an agarose gel that you would use for DNA or RNA, but instead of agarose, it's a polyacrylamide. But the principle is the same. Smaller proteins run farther down the gel, whereas larger proteins stay up near the beginning. Just like with a southern or a northern, we transfer the proteins out of the gel to a membrane, and then here we use our antibodies to probe. And then we can visualize. They could be fluorescent. Oftentimes you use autoradiography. Something like horseradish peroxidase will show up on X-ray film. So they're not radioactive, but you can look at them on X-ray film. And then here, what we can look at is 8 different tissues. It doesn't really matter what tissues they are, probed with the same antibody. Now this is really interesting because what it shows us is that the 8 different tissues, right, one of them doesn't really have much of a band at all, so perhaps that protein is not made in tissue 1. Tissues 2 and 3, the protein is about the same size, right? Because remember, if they're at the same level, they're the same size. So it looks like tissues 2 and 3 process the protein the same way. But then when we look at tissues 4, 5, 6, 7, and 8, the proteins are all different sizes. And so this is really interesting because it could tell us that the rest of these tissues, they do make the protein. I would argue all 7 of these tissues make the protein. But with the exception of these 2 right here, all of the other tissues modify that protein in different ways. Perhaps there's some alternative splicing going on. Perhaps there's some post-translational modifications. But that's what I would say, that these 7 tissues, this protein is being made. What the tissues are doing with that protein, it could be very, very different. And that's something really interesting to think about. That was one of the first things we talked about when class started, is how many genes are found in a human. Anywhere from 20 to 25,000 genes. But scientists have estimated anywhere from 400,000 to 1 million proteins. So how do we get that? Something like this. Alternative splicing and various post-translational modifications.